Previously, in biology, we were going over the structure and function of the nucleus and the endoplasmic reticulum, both smooth and rough. Now, we're going to follow the pathway of the proteins that are made in the rough endoplasmic reticulum. From this figure here, you can see we have the nucleus and the rough ER, and you can see how close they are together. And then from the rough ER, what's called a vesicle kind of pouches off of the rough ER, and then it travels to a structure called the Golgi apparatus, which you can see here that I'm circling. So the Golgi is made up of just a bunch of membranes that are filled with enzymes. We call them cisternae. I'm not really sure I don't really know too many people that call them that, but a cistern is like a sewer, so something to think about. Anyway, the Golgi's purpose is to take the materials that go into it and modify it in some way to allow it to either leave the cell or to be incorporated into the cell membrane. It also will manufacture a certain organelle called a lysosome. So here you can see a couple of representations of the Golgi apparatus. On the left we have the drawing, and you can see it's got a face that is kind of like a receiving end, of like a receiving dock, if you will, and those transition vesicles, or sometimes they're called transitory vesicles, that are coming from the rough endoplasmic reticulum. What they do is they fuse onto that side, and then they get modified as the materials, I should say, get modified as they're a part of the Golgi apparatus, and then they'll come off of what's called the trans side. So it's kind of the exporting side, where the materials will kind of break off in another type of vesicle. In some cases, it's called a secretory vesicle. And then it'll either fuse with the cell membrane or it may be delivered somewhere else. If you look on the electron micrograph, you can kind of, where I have the arrow kind of pointing, you can see where a number of the different vesicles have kind of fused with the Golgi, and it almost looks enlarged. That's because, you've got, kind of these roundish vesicles that are combining with the Golgi apparatus, and then eventually they kind of flatten out. So you can see the different layers of the Golgi. This is a very dynamic organelle because you have additions to one side and then subtractions to the other. So you can see these secretory vesicles here on the bottom that are leaving. So eventually, where these transitory vesicles, where they fuse, eventually they'll become the middle and then the trans side of the Golgi. In addition to preparing proteins and other materials that are in the Golgi for export out of the cell, the Golgi apparatus also makes something called a lysosome. And as you can see from the text, it's a membranous sac of hydrolytic enzymes that can digest macromolecules, whatever that means. So think about like hydrolytic reactions with macromolecule disassembly. So these hydrolytic enzymes, what they do is they can take like macromolecules like proteins, starches, and other types of large biomolecules and break them down into their component parts like amino acids, monosaccharides, mononucleotides, fatty acids, and so forth. So that can be utilized for a couple different things. It can be utilized to break down, say, food that's coming into the cell. It can also be utilized to break down organelles that have kind of broken within the cell. Those parts can be reused. But in some cases, the lysosome can be used to actually kill the cell, which doesn't kind of make sense. Why would cells want to kill themselves? Well, during development, it can actually be very useful. So if you look on the left, we have a tadpole, and then a frog, some froglets, and then a frog. And if you'll notice, the tadpole has a tail, the froglets have a tail, but the frog doesn't. So at some point during that frog's development, the cells in the tail will actually create a lot of lysosomes, okay? And those lysosomes will eventually break open. And remember, they have the hydrolytic enzymes. So the actual lysosomal membrane, is kind of holding those enzymes inside that little package, and it's keeping the cell safe. Once those lysosomal enzymes kind of leave the lysosome and go into the cell, they can cause a lot of damage, which is why the cell actually wants to kind of keep those enzymes in the lysosome. But in cases like with frog development, the cells will make a lot of lysosomes and then allow the hydrolytic enzymes to be released in the cell, basically killing the cell. So that's very useful for the frog's development because it'll basically destroy all the cells in the tail, and then the tail goes away. During human development, you can see here on the right, we have kind of a limb bud in the beginning of a human hand. So the cells kind of along these 1, 2, 3, 4 different planes on the cell, what they do is they do the same thing that the frog tail does. They create a lot of lysosomes, and then those lysosomes break open and allow the enzymes to kind of release just within those cells, then thus killing those cells. Well, that ends up creating our fingers, really. I mean, the bones are in the limb buds in cartilaginous form, but our hand is still just kind of this one shape. The fingers aren't cut, but as throughout the process, you can see, like this picture B here, that the fingers are starting to cut, and then over time you get kind of a fully formed hand. There are some syndromes, though, that some people have throughout development, where this apoptosis doesn't quite happen the way we would imagine or necessarily want to. So I'm going to show you a picture. The next slide is going to show somebody with webbed, what's called webbed fingers. This is called syndactyly. So in this individual, throughout their development, this process of apoptosis didn't quite happen, like I said, the way we want to, and the fingers got webbed. So the lysosome, like I said, is going to be very important for processing food that comes into like a cell. There's a process called phagocytosis, which means cell eating. So when the cell is able to eat like another cell, the lysosome will actually kind of meet the vesicle that's formed through phagocytosis. It'll meet up with that and then fuse with it and then release its enzymes on the cell or whatever else was engulfed, and that will allow the enzymes to break down the the materials, and then the cell can utilize those materials. Say it has, some proteins, it breaks down those proteins, and it'll break them down into amino acids, and then the cell will take those amino acids and make its own proteins. So if you look here on the left, we have phagocytosis, where a cell is entering, or I should say a foreign cell is entering our eukaryotic cell in this food vacuole. Then the lysosome that's filled with those digestive enzymes meets up with it and then breaks down that cell. And then, as previously mentioned, if an organelle is broken or getting worn down, the cell can also recycle those parts. So we call it autophagy. Auto means self, phagy means eating. So it's self-eating. So if you have, a busted mitochondria, a busted peroxisome, the cell can lay down a vesicle or a vacuole around them, and then a lysosome will meet up with it and then digest those down. So here you can see kind of a larger picture of phagocytosis, where we kind of marked the lysosome. Up here you can see the nucleus. I don't see the Golgi anywhere in here. I see a lot of mitochondria. And then here is a really nice photo of autophagy. So you can see this vesicle that's laid down around the broken mitochondria, and then something called a peroxisome. And then a lysosome is on its way to kind of break those down. Once you get engulfed in that vesicle as an organelle, your time is up. Anyway, let's put all those elements together for the endomembrane system. So here we have the nucleus and the rough ER and the smooth ER. Remember, the information from the nucleus is going to leave through the nuclear pores. It's going to go to the rough ER, where a ribosome is going to make a protein. Then that protein is going to get put into a vesicle called a transitory vesicle, and it's going to leave the rough ER, and it's going to go to the Golgi apparatus, all right, where it will fuse on the cis side. Enzymes inside the Golgi will modify the proteins or whatever else is in there for export out of the cell. All right. Eventually, it will leave the trans side of the Golgi apparatus once it's been modified into a secretory vesicle, and then it will fuse with the plasma membrane or the cell membrane, and those materials will leave the cell. But wait, there's more. While that kind of puts it all together, going from the nucleus all the way to the Golgi and out of the cell, there are some other kind of odds and ends to the endomembrane system that don't necessarily kind of play in that nice kind of linear fashion. So we have some things called some vacuoles. So a plant cell will have what's called a central vacuole, and in some cases it can be up to about 90% of a plant's volume, which is incredible. In some other kind of microbes, we have what's called a food vacuole and a contractile vacuole. So food vacuoles are going to occur even in like us, where we do phagocytosis, and then whatever the vesicle or vacuole that comes into the cell is filled with food. That's food vacuole. But in some microbes like Paramecium, they have this thing called a contractile vacuole, where if it has too much water in it, it'll actually make this little vacuole and pump the water out that way. If you look here, we have an artist's conception of a central vacuole, and then an electron micrograph. So the central vacuole in a plant can be important because it's going to hold materials, and these could be holding starch, it could just be holding water to kind of create a pressure inside of the plant. It could be holding waste products. It could be holding pigments, like for instance in flowers. In the cells of the flower, the central vacuole is going to be holding that pigment, keep giving it its color.